Welcome to the second of 4 webinars that the National Cannabis Prevention and Information Centre will be hosting in October and November this year. My name is Dion. I'm a research officer here in the clinical training team, and I'm going to be introducing you today to our presenter, Dr. Tim Karl. I'll also be moderating the question time at the end of the presentation, so please type through any questions you would like answered by Dr. Tim Karl while the talk is in progress, and I'll do my best to make sure that these are addressed at the end. So today's presentation by Dr. Tim Karl is on the impact of cannabis constituents on schizophrenia and Alzheimer's disease, a mouse perspective. Tim established his own behavioural neuroscience group at Neuro, which is Neuroscience Research Australia, and his research focuses on the neurobehavioural consequences of gene-environment interactions in rodent models for brain disorders such as schizophrenia and Alzheimer's disease. I hope you enjoy today's presentation. Tim, over to you.
Thank you, Dion, for the introduction. So what I will try to do today is give you some examples how cannabis is evaluated and investigated in a medical research context. I would like to stress up front that I'm not talking about cannabis as the drug which is available legally in Australia on the drug market, but we are talking about specific cannabis constituents, so certain components within the cannabis plant which we are investigating regarding their medical purpose or their medical value. And I also have to stress all the work I will present today is in mouse models, so it's preclinical research. I will try to cover some of the potential regarding translational value of our research as well, but all the data I'm showing you is based on mouse model research. So to start, I will give you a little bit of a background about cannabis. That will be very brief, and then there are basically two degree-related sections. One is on the effects of cannabis or cannabis constituents in schizophrenia, and the other one is on cannabis in Alzheimer's disease. So as I already stressed during the introduction, cannabis is actually very complex plant. Cannabis sativa makes up to 70 different plant constituents, and these constituents are called cannabinoids. One of these constituents is called delta-9 tetrahydrocannabinol, and that is THC. That's probably better known in the public. This is the main constituent of the plant. It is psychoactive. It is cognitive impairing, so it basically impairs your learning and memory performance. And this is basically the component which makes you stoned, or this is the component why people actually use cannabis as a recreational drug. However, there is another cannabis constituent which is called cannabidiol or CBD. This can make up to 40% of the cannabis concentration, and this constituent is non-psychoactive, so it has no impact on the mental state if you are treated with this component, and I will get back to this in a moment. Just for clarity, there are a lot of other cannabinoids in the cannabis plant, such as THCV, CBDA, CBG, and others. But I will not cover any of those, as most of the medical research done on cannabis focuses around THC and CBD. So very basic introduction into cannabis biology. There are two receptors in the brain, or two docking stations in the brain, the cannabinoid receptor 1 and cannabinoid receptor 2, and they mediate most of the functions of cannabis and of its constituents. Importantly, what you can see here at the bottom, this is a slide of a mouse brain, and what you can see is that all these yellow and red highlighted areas basically give you an indication of the high expression of cannabinoid receptors in the brain. So it is one of the most abundantly expressed receptors in the brain. And obviously there has to be a reason for this, and I will try to cover this further on in my talk. It is also established by now that they are so-called endocannabinoids. So within the brain, there are substances which are released by synapses, and these substances actually activate these receptors. They are called endocannabinoids, and again, this suggests that the endocannabinoid system and these cannabinoid receptors have an important biological function in the brain, potentially for brain development, etc. Just to cover some of the legal and society-related aspects of cannabis and cannabis research, It is a recreational drug, and although there are discussions around its potency regarding developing dependency, it has been shown that at least a certain level of dependence can be developed when you are a heavy cannabis abuser. It is not as addictive as some of the other drugs, but as you can see here, cannabis definitely can lead to cannabis dependence. And this is actually a growing problem. This is a larger, a rather old figure from 1993 to 2003, but this gives you an indication how many people actually suffered from cannabis abuse when they were actually admitted to certified substance abuse treatment facilities in the US. And within 10 years, this percentage actually had doubled. So cannabis as a recreational drug is a growing problem. On the other side, there are lots of reports. Some are really just incidental reports by individuals. Others are based on medical research, and I will try to cover some of this today, that some aspects of cannabis, and these are the different cannabinoids expressed in the cannabis plant, might have some value for medical research. And one of the most interesting targets at the moment is probably CBD, and I will cover this in quite some detail over the next few slides. So the first focus will be the role of cannabis in schizophrenia. Just to give you a quick introduction to schizophrenia, and I'm not a psychologist, I'm really a behavioral neuroscientist, so I keep this very brief. The cause of the disease is genetically and environmentally linked. So they are genetic, they are genes basically. If you have a mutation in these genes, that increases your risk to develop schizophrenia, and there are also environmental factors such as obstetrical complications or drug abuse, which increase your risk to develop schizophrenia. The prevalence of schizophrenia is actually 1% over the lifetime, so that means in Australia around 2,500 people per year develop schizophrenia. The theory behind the development of schizophrenia is based on the new developmental theory. This states that these risk factors, genetic and environmental risk factors, and in all likelihood actually in interaction, impact on neurotransmitter systems in the brain, and the alteration of these systems actually change brain development and maturation early in life, and this eventually results in the development of schizophrenia-like symptoms. So how does a human phenotype look like? This is a photo of John F. Nash. He was a Nobel laureate for economics. His life history is actually featured in the movie A Beautiful Mind, which you might know as the main actor was Russell Crowe. He suffered from schizophrenia later in life, and the symptoms which are listed here for schizophrenia are not all inclusive. That means some patients will show some of the symptoms and other patients might show other symptoms. We normally distinguish between positive, negative, and cognitive symptoms. Positive symptoms are things like hallucinations, suffering from delusions, and thought disorder. This is normally summarized as psychosis. There are negative symptoms, which are defined as social withdrawal, lack of motivation, and poverty of speech and olfaction. And then there are cognitive deficits you can find in schizophrenia patients, which basically means you have an impaired learning and memory performance, you show reduced attention, and you have something which is called sensorimotor gating impairment. And I will explain in a while what this actually means, because that is one of the features which we also test in animals. Some other facts about schizophrenia. Normally the onset is at late adolescence to early adulthood. It is normally earlier in men than in women. However, late onset is possible in women, and this is potentially based on the drop-off in estrogen levels after menopause. There are other gender differences reported for schizophrenia. Men normally show more pronounced negative symptoms and cognitive symptoms, whereas they are less susceptible to the side effects of antipsychotic treatment. Over the last 10 years, the field probably has started realizing, although this is still hotly debated, that in all likelihood we are not looking at a single disorder, but rather at a number of discrete syndromes. Part of the reason for this shift in opinion on schizophrenia is based on the fact that there are so many genetic risk factors out there and so many different environmental factors, and also that not every schizophrenia patient shows all the symptoms I just explained to you. So in all likelihood, we look at different subsets of disorders which are potentially related to each other. And this will be important when we talk about how mouse models can help in schizophrenia research. So regarding the therapy, there are typical and atypical antipsychotics. Unfortunately, they are effective in as little as 20% of the patients. Quite often you have to use combinations of these antipsychotics, and they show very limited effects on negative symptoms. So that is one of the reasons that patients actually show little compliance with the treatment designs developed for them. And this is also due to the fact that most of these antipsychotics have quite severe side effects, and those include motor impairment, sexual dysfunction, and weight gain. And as I said, that the onset is normally at late adolescence or early adulthood. You can imagine that these side effects are actually taken on quite severely by the people suffering from schizophrenia. So how do mouse models help us with schizophrenia research? Obviously, when we look at a mouse model, and some people use rat models as well, we don't assume that these animals are actually schizophrenic. First of all, we are not able to test hallucinations or delusions in these animal models. Secondly, we are not even sure if these animals experience hallucinations or delusions. However, these animal models can help to investigate certain aspects of schizophrenia, and that's why we call them a translational paradigm for what we would call an endophenotype. So you, for example, can look at the cognitive deficits in schizophrenia. You can develop a mouse model for this, and then you just focus on the cognitive performance of these animals, and therefore might understand a little bit better how certain genes or certain environmental factors impact on cognitive domains when you are suffering from schizophrenia. How do we rate these models? There are basically three criteria for schizophrenia mouse models. They are called phase, construct, and predictive validity. And to keep it simple, phase validity just means that you have a phenomenological similarity between the behavior your animals show and the human symptoms. So if I just explained to you that schizophrenia is defined by cognitive deficits, you would expect that your mouse model shows deficits in learning and memory as well. A second criterion is construct validity. That means that there is a similarity between the underlying mechanisms for the behavior shown in these animal models. So if, for example, there's a risk gene for schizophrenia in humans, and if you have a mutation in this gene, this increases the risk to develop the disease, then a model has construct validity if you modify this gene in the mouse as well. And lastly, you hope for predictive validity. That means if your model shows behavioral symptoms which are relevant to schizophrenia, you would hope that with antipsychotic treatment, some of these symptoms can be reversed. So how does such a human phenotype translate into a mouse phenotype? We normally assume that a mouse model has at least some validity for schizophrenia if these animals are hyperactive, they show impairments or deficits in learning and working memory, they show deficits in sensorimotor gating, they show reduced social interaction, so they interact less, they show less social behaviors, and some of these symptoms should be reversible by antipsychotic treatment. So why do we investigate actually the effect of cannabis in schizophrenia? It is very well established that there is a history of heavy cannabis abuse in schizophrenia patients. Up to 45 to 50% of schizophrenia patients have a history of cannabis abuse. Furthermore, it has been shown that cannabis consumption can increase the severity of positive symptoms and the risk of psychotic relapse. And finally, some studies suggest that chronic cannabis abuse in adolescence, that is important, it has to be at a critical time of your brain development. Chronic cannabis abuse in adolescence increases your risk to develop psychosis by twofold. Having said that, that is a very relative measure. If your general risk to develop schizophrenia is 1%, then increasing your risk by twofold means your relative risk to develop schizophrenia is 2%. If you have a family history of schizophrenia, and let's assume your genetic risk to develop schizophrenia is 15%, then smoking cannabis might actually increase this risk to 30%. So it really depends on the context in what way cannabis impacts on your risk to develop the disease. Why is it such a complex interaction between cannabis and schizophrenia? First of all, and I mentioned that already, cannabis sativa is a mixture of up to 70 different cannabinoids. And just to stress again, one of the major constituents of the plant is cannabidiol. This cannabinoid actually lacks psychoactivity. And furthermore, it has been shown that it can block the effects of THC. So if you look at this figure here, you can see psychotic symptoms induced by THC. As I said, THC is psychoactive. If you are treated with THC, you can see that your psychotic symptoms increase over a period of time. However, if you are pre-treated with CBD, this actually blocks most of the psychotic effects of THC. So dependent on what cannabis plant you're actually smoking, what strain is used, and dependent on how much THC and CBD is expressed in this strain, you might suffer from more or less psychoactive effects of cannabis because CBD will block some of the effects of THC. Furthermore, there are pharmacological models of psychosis. So you can, for example, use ketamine, and ketamine induces an acute psychotic-like state. And this is shown here. This is when people are treated with ketamine, they show a depersonalization factor score of a certain degree. If these people are actually treated at the same time with CBD, some of these psychotic-like effects of ketamine are actually reduced. So you could argue that in humans, CBD has to a degree an antipsychotic-like effect, or at least it blocks some of the effects of ketamine. Furthermore, the same has been done in animals. When you use dexamphetamine, which is another psychotropic-like drug, animals normally show hyperactivity. So they become more active. They run around more in an arena. If you treat these animals with a very high dose of CBD, what you can find is that this hyperactivity is actually reduced. So again, CBD has potential to minimize or reverse some of the psychotic-like effects of dexamphetamine treatment. Secondly, when you talk about cannabis and schizophrenia, you have to be aware that the potency of cannabis available on the drug market actually has changed over the last 30 years. This here is a figure which shows you the THC, so the psychoactive component, the THC content of cannabis in 1996, 98, and 2005. And what you can see is that the THC content of cannabis has significantly increased over these 10 years. So people are nowadays, when they are abusing cannabis, are exposed to much more THC than they have been in the past. However, if there would be a clear causal relationship between cannabis abuse and schizophrenia, you would expect that if THC content is increased in cannabis, that therefore more people develop schizophrenia. The prevalence of schizophrenia across the population hasn't changed in the last 30 years. It is still around 1%. And lastly, research shows that around 90% of cannabis abusers will never experience any psychotic symptoms. It also has been shown that the endocannabinoid system is involved in schizophrenia. So I told you about the two receptors for cannabinoids, CB1 and CB2, and depending on in what brain area you are looking and what type of schizophrenia patients you are looking, you can find that these receptors are up or down regulated. What all this suggests is that in all likelihood, cannabis is a component or cumulative cause of schizophrenia, which means that it interacts with other risk factors for the disease to actually then increase your risk to develop the disorder. One of these gene-cannabis interactions is shown here. So this is a figure which shows you the percentage of 26-year-olds which develop schizophreniform disorder. And these people have, regarding one particular gene, a different setup. So there are 3 different setups available for this gene. And you can see that only if you have a particular type of this gene, the VALBAL type, you actually increase your risk to develop schizophrenia if you abuse cannabis. So this gene, it's called catechol O-methyltransferase, or potentiates the influence of adolescent cannabis use on adult psychosis. So this suggests that people with genetic predisposition for schizophrenia, so who have a family history of schizophrenia, might be more vulnerable to the effects of cannabis. There is a similar interaction known for an environment cannabis scenario. So as I outlined before, if you abuse cannabis, during adolescence and you're a heavy cannabis abuser, that increases your risk to develop schizophrenia by around 2-fold. If you have been abused as a child, research suggests that this also increases your risk to develop schizophrenia later in life, and it's probably an increased risk around 20-fold. If you have been abused as a child and therefore you develop psychotrauma and you abuse cannabis later in life, your risk actually increases by 200-fold. So that's an environment-cannabis interaction. Our research wanted to know if such an interaction is also true for another schizophrenia risk gene, and this risk gene is called Neuregulin 1. So Neuregulin 1 has been shown to be associated with schizophrenia, as in that a mutation in this gene can increase your risk to develop the disorder, and that has been established and confirmed across a large number of different cohorts worldwide. The gene is critically involved in how the brain responds and adapts to the environment. It is expressed in brain areas which are relevant to schizophrenia, and schizophrenia patients show different expression of this gene. So this suggests that this gene is actually important for schizophrenia. So we in our lab have a mouse model which has a mutation in this gene, and this mutation is similar to what has been found in schizophrenia. So these animals show construct validity and are therefore a valid model for schizophrenia research. They are also hyperactive, they show cognitive impairments, and they show social deficits. Therefore, they can be used for research into schizophrenia. And we wanted to know how these animals respond to cannabis, or to be more precise, to particular cannabinoids. And we focused on THC, on CP55940, which is a synthetic cannabinoid, but it has the same characteristics as THC. And we wanted to see if cannabidiol, or CBD, might have antipsychotic-like potential in these animals. So that you understand the next few slides, I will give you a very brief introduction in some of the tests we have run. So the one test we are using is the open field test. This is basically just an open arena. Animals run around in this open arena, and you can measure their locomotion. And you can also test if these animals are anxious, because animals which are stressed in this test don't explore the central area. They will stay in the periphery and just explore the peripheral area of this test. A second test is the Sensorimotor guiding or Prepulse inhibition test. And as I mentioned before, schizophrenia patients show a deficit in Prepulse inhibition. And I will explain this test in a little bit more detail, because this is actually a test which is run exactly in the same fashion in humans as in animals. So it has high translational value. So Prepulse inhibition is defined as a reduction in the startle response caused by a preceding prepulse. So what does it mean? Assume you sit in a sound-insulated room and you're exposed to 120 decibel tone stimulus. You will show a startle response to that, and this is shown here. However, if the startle tone with 120 decibels are preceded by a prepulse, which in itself doesn't induce a startle response, you will show a reduced startle response, because the prepulse basically inhibits your startle response to the actual tone pulse. So you have a reduction in the startle response caused by the preceding prepulse. So this is basically a filter mechanism to avoid sensory flooding. This is a good thing. You want to have this. This filter mechanism is very important. However, this is deficient in schizophrenia patients. And it also has been shown that psychotropic drugs actually impair this prepulse inhibition. The beauty of this test is that we can run exactly the same protocols in humans as in animals. So humans are tested in a room, and we test their startle response by measuring the eye blink response. In animals, they sit in little chambers, they get exposed to these tones, and we measure their whole body flinch amplitude. The protocols we are running are pretty much identical. The third test we use is the so-called social interaction test. So you basically just put two mice together, and you want to check what social behaviors these animals show and how much time they spend on social interaction. And this is a model for social withdrawal to again model some of the symptoms of schizophrenia. So in the first aspect, we treated our animals acutely with THC. So they got one shot of one particular dose of THC, and 30 minutes later we tested them in different behavioral paradigms. I will just give you a few examples for what we found. The first thing we found was that these are always our control animals, so they are healthy. And on the right side you see our Neuregulin mutant animals, so they have a modification in the gene which is relevant to schizophrenia. And what you see here with the white bars, these animals have been treated with vehicle, so they don't have THC on board. And these animals are hyperactive, because that's what we would expect of a schizophrenia model. However, when we treat them with THC, and we treat them with 2 different doses, you can see that actually the hyperactivity disappears. So THC has a locomotor suppressive effect. However, this is already significant or relevant for animals which are schizophrenic-like at a much lower dose than in animals which are wild-type-like. So you could argue that these animals, when they have this mutation, are more susceptible to the effects of THC. We also looked at Prepulse inhibition, and I just explained to you what this test means. So remember, increased Prepulse inhibition is a good thing. Or the other way around, from a schizophrenia mouse model, you would expect that it has a decrease in Prepulse inhibition. And that's what we see when the animals are treated with vehicle. They show less Prepulse inhibition than the control animals. However, and that was quite surprising, when animals are treated acutely, and that's important, and I get back to this when I explain the next experimental steps. When the animals are treated with THC, they actually improve their Prepulse inhibition. So acutely, THC had a beneficial effect on these mice. And this might be in line with the quite controversially discussed self-medication hypothesis of schizophrenia, where some, not all, but where some schizophrenia patients report that initially when they abuse cannabis, they actually feel better. This beneficial effect disappears over time, and I will show you data for that which are similar in our mouse model as well. But initially, cannabis actually might have some positive effects, and that might be one of the reasons that people start abusing cannabis when they are suffering from schizophrenia. We also looked at the brains of these animals, and without going into too much detail, we just wanted to see how much THC activates the brain. And what we found was that only in our Neuregulin mutant animals, they respond with an increased activity in certain brain areas when they have THC on board. So again, that suggests that these animals are more susceptible to the effects of THC than normal healthy control animals. In the second step, we wanted to look at chronic THC administration, or actually CP challenge in this case. As I said, CP is a synthetic cannabinoid, but it has the same characteristics as THC. So we can kind of use it as a THC equivalent. So we treated animals for 15 days with CP and tested them every second day in certain behavioral tests. And again, I just give you a little example for some of the findings we found in this test. This is again the locomotion of these animals in the open field test. So how much do they run around? We find that our mutant animals, when they are on vehicles, so don't have THC on board, are hyperactive, and this was to be expected. And we also find that on day 1, all animals show with a suppression in locomotion when they have THC on board. Again, that's expected. THC has a sedative-like locomotor suppressive effect. However, what is known for cannabis is that normally animals as well as humans develop a behavioral tolerance to the effects of cannabis, which means that if you keep using the same amount of cannabis, you get used to this amount and you show less of an effect, which normally results in increasing dosage over time. So this is called behavioral tolerance, and that's what we generally see in all our animals which are treated with CP. These animals, slowly over the next 7 days, actually get back to control levels and don't show a suppression of the locomotor activities anymore. However, this happens much more readily in our schizophrenia mice. So again, you could argue that these animals are more sensitive to the effects of cannabis or to the effects of CP. We also looked at sensorimotor gating again. This here is day 1. That basically recapitulates what I showed you in the first experiment. The animals actually show, our Neuregulin animals show an increase, that they have a deficit in Prepulse inhibition, and they show an improvement in Prepulse inhibition when they have CP on board. However, and that is important also in regards to the self-medication hypothesis, this beneficial effect disappears once you treat them for 7 and 15 days. We looked at the brains again, and to cut the long story short, we again find that only our mutant animals actually show an increased brain activity after chronic exposure to CP, suggesting that these animals are more susceptible to the effects of CP. Lastly, we looked at how cannabidiol actually impacts on our mice. And for that, we treated them for 3 weeks with cannabidiol and again tested them in certain behavioral paradigms. We first looked again at locomotion. What you can see here, these are our control animals, these are our mutant animals. What you can see is that our mutant animals are hyperactive when they have vehicle on board, so no CBD, compared to the control animals. They are still hyperactive when they have a low dose of CBD on board, but the hyperactivity actually disappears when they have 50 milligrams or 100 milligrams of CBD on board. However, this is statistically not relevant because CBD has a slight effect in our control animals as well. So this slide in itself, or these data in themselves, don't suggest that CBD has an antipsychotic-like potential. However, we also looked at social interaction. So again, we looked at the time the animals spent socially interactive with each other. And our mutant animals have a deficit compared to the wild types, so they show less social interaction. But this can be reversed with a high dose of CBD. So in this regard, you would have to argue that in this mouse model, CBD has an antipsychotic-like potential. So to summarize this part of the talk, we found an elevated sensitivity of Neuregulin mutant mice to acute THC treatment, a more rapid development of tolerance to the chronic effects of CP. As I said, that's an equivalent to THC. And CBD reversed some of the schizophrenia-relevant deficits, and I've shown you here only the data for the social deficit, which is one of the negative symptoms. So to conclude this aspect of this part of the talk, Neuregulin 1 seems to be involved in the neurobehavioral vulnerability to cannabis exposure, and CBD might have potential to reverse particular schizophrenia-relevant behaviors. Importantly, and I'm aware that we have done this only in mice and that we only did this in one particular mutant mouse model, importantly, this research was followed up on by clinicians. And what these people found was that actually a mutation in the Neuregulin 1 gene impacts in humans on their risk to develop cannabis dependence. So this shows you some translational value or validity of our mouse model research in the human condition. So coming to the second part of the talk, and this is how cannabis or a certain cannabis constituent might be relevant or important for Alzheimer's disease. I keep the introduction to Alzheimer's disease as brief as I can. It is basically characterized by 2 pathological hallmarks, and they are called amyloid plaques and neurofibrillary tangles. So you find increased levels of those in Alzheimer's disease patient brains. Further pathophysiological features of Alzheimer's include increased neuroinflammation, increased neurotoxicity, increased oxidative stress, and neurodegeneration. Currently, there are treatment options available which only target very particular aspects of Alzheimer's disease. And unfortunately, all of these treatments have limited efficacy, so they only treat mild to moderate symptoms, they don't have any long-term effects, and they definitely do not stop or reverse the development of Alzheimer's disease. Furthermore, they actually develop quite severe side effects, including hallucinations, dizziness, gastrointestinal complications, and encephalitis. So there is definitely a need for new treatment options, and the field nowadays probably agrees that you should use a multimodal drug approach, which means you should use a drug which targets different aspects of Alzheimer's disease at the same time, and you should have a drug which ideally you can give early on and long term, because you obviously want to prevent the development of Alzheimer's disease as early as you can. There is some indication, limited evidence, but some indication that the endocannabinoid system is involved in Alzheimer's disease. The field still discusses if it has a positive effect or a negative effect, but it definitely has been shown that, for example, one of the 2 receptors for cannabinoids, the CB2 receptor, is differentially regulated in Alzheimer's disease patients. So you can find up and down regulations of this receptor, which indicates this system might play a role of some sort in the development of the disease. As I said before, cannabis has been considered as a potential target for medical research, and I will explain you in the following why we have focused on CBD for Alzheimer's disease research. So the one important thing about CBD, apart from that it's not psychotic-like, is that it doesn't impair cognition. This is obviously highly important when you think about Alzheimer's disease, where people actually suffer from cognitive impairment. It also has been shown in the clinic, so in humans, that it is well tolerated and it doesn't seem to have any side effects. Furthermore, there is already a pharmacological compound out there which is called Sativex, which is an oral mucosal spray which contains THC and CBD at a ratio of 1:1, and this is approved, for example, in Canada for the treatment of multiple sclerosis. It doesn't stop the development of the multiple sclerosis, but it helps with some of the symptoms. And for Australia, it's important to know that CBD has been registered by the Australian Therapeutic Goods Administration for the use in people. It is not legal yet to use CBD in Australia, but at least generally speaking, it might be at one point become available for clinical trials and for further research into its potential for medical research. So what are the biological characteristics of CBD, beyond the ones I've explained to you when we were talking about schizophrenia? CBD has been shown to be neuroprotective, it's immunosuppressive, it is antioxidant, and anti-inflammatory. And if you remember the slide I showed you at the beginning of the Alzheimer's disease part, all these areas actually are impaired in Alzheimer's disease. So we thought it is actually not that unreasonable to hypothesize that CBD might have therapeutic potential for Alzheimer's disease, as it seems to target different impairments or different aspects of the disease. And there is some in vitro evidence. In vitro evidence basically just means you have cell models for Alzheimer's disease. They are very limited models. This is why you always at one point have to go into the in vivo models and use, for example, rodent models. But they give you a first indication what CBD actually might be doing. And it has been found that CBD decreases neurotoxicity and oxidative stress. It has also been shown that it actually reduces or inhibits tau hyperphosphorylation, which is one of the reasons for the buildup of neurofibrillary tangles. It reduces neuroinflammation, and it also reduces A-beta production, and A-beta production is a precursor for the development of cynical plaques. So in vitro, it seems to be doing something good to two of the pathological hallmarks of Alzheimer's disease. Does this hold up in vivo? Before we started with our research, there was actually only one study which had looked at CBD in vivo. There are pharmacological models of Alzheimer's disease, and these models have found that CBD actually prevents cognitive impairment, which is shown here, and it also decreases neuroinflammation. So again, some preliminary evidence that CBD might be relevant for Alzheimer's disease. So what we did, or what our aims were, we have animal models available which develop Alzheimer's disease. They are transgenic, so genetically modified animals, and we wanted to see if CBD is able to reverse Alzheimer's disease relevant symptoms once animals have developed Alzheimer's disease. And we also wanted to see if it actually prevents the development of Alzheimer's disease-relevant symptoms. This was done by my PhD student, David Cheng. So we used this so-called double transgenic mouse model. It has genetic modifications to actually develop Alzheimer's disease. The animals develop cynical plaques, and they show cognitive deficits, which is important because that is basically what we use as a readout to test the effectiveness of CBD. As I said, we had two experimental designs. One was to see if CBD treatment for 21 days after the development of Alzheimer's disease actually can reverse the cognitive deficits. And the second one was giving animals orally CBD. So they were trying to use pellets every day. They got little pellets with CBD or vehicle. And they were treated for 8 months with CBD. And then we tested if these animals actually didn't develop Alzheimer's disease-like symptoms, and we also looked at the brains of these animals. Again, I explain you briefly what tests we use, and they are pretty straightforward, so it shouldn't be too complex. So the one test is the novel object recognition task. In this test, animals are first exposed to two identical objects. And then the second time around, we expose them to a familiar object and to a novel object. If the object recognition memory of your animal is intact, it will recognize that this is a novel object and then spend more time with the novel object. We also use the test for social recognition memory, where animals are first exposed to one mouse and then the second time around to the familiar mouse and a novel mouse. And again, you would expect that your mouse, if the social recognition memory is intact and can recognize the novel mouse, it spends more time with the novel mouse. And lastly, we use the G-spot task. This is not the G-spot task, but that comes up when you Google it. So it shows you that sometimes Google is not the best way to search for a certain context. This is how the G-spot task looks like. It is basically a platform. Animals are food deprived, and in one of these wells we have a little food reward hidden. And animals learn over time to find this food reward. And then at one point you take the food reward away, and animals will then, if they have memorized the location of the food reward, spend most of their time next to the well because they expected it to be baited with some food, and this is your way to test their spatial memory. So first the study and the results when animals had developed Alzheimer's disease symptoms, and we then treated them with CBD. These animals, as expected, when they don't have CBD on board, show a deficit in object recognition. So they don't have a preference for the novel object. If we treat them with CBD, we can reverse this. So the animals actually then can recognize the novel object again. When we test animals in regards to the social preference test, we first check that they actually don't have an aversion to investigate a mouse. And all these animals show a clear preference for the mouse over the empty chamber. And we then tested their social recognition memory, and what we found was that the animals, when they are affected by Alzheimer's disease, they lose their recognition memory. So they don't have a preference for the novel mouse. However, if they have CBD on board for 3 weeks, this can actually be reversed, and the animals again show intact recognition memory. We also did the preventative study, as I said, long-term treatment with CBD before the development of Alzheimer's disease symptoms. And what we found here, again, in regards to the social recognition memory test, animals develop a deficit if they get vehicle pellets, which don't contain CBD. However, if they have been treated with CBD for 8 months, these animals show intact social recognition memory. Furthermore, if you test them with a spatial reference memory test on the spatial memory task, they don't have a preference for the food reward location. But if they are exposed to CBD for 8 months, they actually show a normal spatial memory ability. We also looked at the brains because obviously we were quite excited based on our findings, and looked at amyloid load, which is again a precursor for the development of senile plaques. Interestingly, the animals didn't show any effect of CBD, so they had the same levels of plaques or amyloid loads in 2 areas of the brain. We then thought perhaps CBD impacts on the oxidative stress in these animals. But again, CBD had no impact on the oxidative damage in these animals. Last, we looked at neuroinflammation, and there was at least a trend for our Alzheimer's disease animals to have increased neuroinflammation, which is shown here. And CBD was able to reverse this effect back to control levels. So in that regard, it had a beneficial impact. So to summarize, we found that CBD was effective in reversing and preventing cognitive impairments in Alzheimer's disease transgenic mice. And the effect of the preventative CBD treatment on social recognition might be mediated by anti-inflammatory properties. So thinking about Alzheimer's disease and the clinical situation, CBD might potentially be beneficial for various cognitive symptoms. The one is social withdrawal, and I have shown you that to a certain degree already during the schizophrenia talk. Also in regards to the fact that Alzheimer's disease patients show difficulties in recognizing faces and also poor object recognition. And long-term administration of CBD, remember we treated the animals for 8 months, might have preventative properties for Alzheimer's disease. Just to again go back to, is this actually in any way relevant in humans? working on mice normally means that you really don't interact that much with the clinical population, with relatives of schizophrenia or Alzheimer's disease patients, etc. However, based on recent publications, our recent publications on CBD and Alzheimer's disease, we actually received some media coverage by the Sydney Morning Herald and also a news blog at the Alzheimer's Association Australia. Based on this coverage, I received, probably for the first time in my life, quite a few emails from people who are affected by Alzheimer's disease, or actually, to be more precise, by relatives of Alzheimer's disease patients. And I'll just give you two examples. And please be aware there are obviously certain legal issues with these examples. So one is from Australia, where THC and CBD is illegal. The other one is from the Netherlands, where it's actually legal to abuse cannabis, and where you therefore also have very readily access to particular oils which, for example, only contain THC or CBD. So the person in Australia produced a CBD tincture and it had 15% of CBD and reported that his father, who is suffering from Alzheimer's disease, showed an increase in alertness, showed improved memory function and a brightness to his thinking. He was calmer and less aggressive. A woman in the Netherlands actually organized, based on our publications, without my input, because I obviously would have to express to her that this is all mouse model research and it hasn't been tested in the clinical situation yet. It purchased CBD oil as well. And she reported that her mother actually needs much less help for getting dressed. She does make her own bread for breakfast and recognizes me. Sometimes she mentions things from a couple of days ago which she hasn't done for months. She recently actually sent me a YouTube video where her mother started playing the piano again, which she hasn't done in years. These are just case reports, but it gives you an indication that there might be some relevance in our mouse work for the translational aspects and for the human sector. So as an overall summary, the effects of cannabis on schizophrenia are complex, and that's obviously based on the over 70 different cannabinoids in the plant. CBD might actually provide potential for the treatment of negative symptoms, and gene-cannabis interactions must be considered in human studies. That's really important when you think about epidemiological studies, where they try to investigate a link between cannabis and schizophrenia. These people actually should be screened for COMT, but also for Neuregulin 1, because these genes will impact, or mutations in these genes will impact on how the people actually respond to cannabis. And CBD appears to be a promising target for Alzheimer's disease therapy. That was the second part of my talk. So in general, to conclude, I think that more research into the effects of cannabis constituents, and that's important. I'm not talking about cannabis, I'm talking about constituents, is absolutely necessary. So I would thank my collaborators at the University of Wollongong, especially Brett Garner, who was heavily involved in the Alzheimer's disease-related side of things. I also would like to express my gratitude to Jonathan Arnold. He's a cannabinoid researcher at the University of Sydney, and he opened up my interest into the field of cannabis-schizophrenia interactions, and the Schizophrenia Research Institute, which was actually very heavily involved in funding this research. You might, or you can, I think, imagine that it's sometimes hard to actually receive funding from governmental bodies when you start looking at cannabis and potential preventative or positive aspects of the plant. Other funding bodies are mentioned down here, and these are some of my team members who were heavily involved in the work. Animal model research is actually very time consuming. I'm very grateful that these people did very long hours in the lab to actually generate all this data. I would like to thank you for your attention.